All right. Well, thank you very much for all that information about Drosophila and embryos. I think I want to ask a question about what types of things did you do with the Drosophila in the cures? What questions did the students ask? What were some of the results? Because I know that I believe that you did this both in a smaller cure and in a full semester cure. And you also did it with first year students and upper level students too. And there were some questions in here about upper versus lower level students. Should the students be choosing their own project? Should the professor be choosing the project? And those are some questions that we didn't get to with Dr. Brownell, but I think that this is a great time to kind of talk about them. So that was a lot I just asked you to do.
That's right. I will try to tackle all of that. Um, so the techniques that I just demonstrated were for the 4-week mini cure that I taught with first year students, most of whom had little to no lab experience. They were only in the lab for 4 weeks. The rest of the time was spent in the classroom learning some research techniques, some introduction to ASU resources and such. Um, but since we had such a limited time in the lab, I really had to restrict just how much freedom I gave them in the laboratory. So in lecture, they learned about doing literature searches, and I presented the question to them about, how do different nutritional supplements influence fly fertility? And they had to go online and find articles that addressed that. And many of the students were able to make this very personal because they thought about home remedies that they knew about and they wanted to see, does this affect the flies? Um, one group was really interested in energy drinks and how that might affect fertility. So they really got excited about different things that they could add to the food. Um, and they had to back that up with the literature and figure out what they would expect to see. And then when they got into the lab, then they could use the protocols for those different techniques that I showed, uh, and simply look to see if there's a difference in egg lay from one treatment to the next. So for each group, they were able to do 2 different treatments with 3 vials each. I did have them work in groups, but that seemed to work out pretty nicely. So uh, there was a lot of prep done ahead of time. So for making the fly food, I mixed up a lot of the dry components ahead of time, so that all they had to do was add, whatever their additive was, and water, and then cook it, add it to the vials, and then they were good to go. So I think there was a lot more beforehand prep for the mini cure than for the larger semester long cure that I taught. So for the semester long cure, rather than just focus on fertility, which is what I set out to do, we actually kind of ended up focusing on diet and a whole lot of other things. So this class, I had a couple students who had little to no lab experience. I had some that had done quite a bit more. I had some students that were fully online. So I had quite the mixture of individuals to try to work with. Um, so with this group, I did have them collaborate via Zoom. So I did have hybrid groups with some online, some in person. They worked via breakout rooms that I could just kind of hop into to see how they were doing and make sure things were going smoothly. Um, but I gave them a lot of freedom with what to do. And I started with those same basic techniques of, this is how you can make the food, this is how you could identify the flies. And they pretty much took it from there. So I had one group that looked at the effects of spinach extract on learning in flies. So they found an assay to do online, and they built the contraption necessary for the decision making for them, but they really got excited about that. For the online group, they looked up a lot of data sets on changes in gene expression associated with different diets, and they really wanted to focus on identifying how different diets affected lifespan. And so they focused on looking for genes associated with lifespan that were altered um, in response to different diets. So we got a little bit of everything in that class. I do see a question in the chat that about the generation time of flies. So you can go from egg to adult in as little as 10 days, which makes it really good for a cure, I think, and lends itself well to in-class experiment. For my semester-long cure, we did meet twice a week, which worked out well for a developmental type cure, because students did have to come in multiple times just to check on the progress of their flies, to count embryos, and just make sure things were progressing appropriately. So I think I got everything you were asking.
Jen, can you put your mic down just a little?
Oh, yes.
Okay.
Thank you.
Airline pilot.
Yes, the exits are here and here. So I think I got everything that you were asking about.
So what, with Drosophila, how did you when you say you studied genes, what do you mean by that? How did you study those genes? Did you do northern blots? I'm dating myself right now.
So the folks that were online, what they did is pour through the literature and identify transcriptome data sets that were publicly available. They would get those data sets, find the ones that were differentially expressed, and look for overlap between any of those lists. So they were really focused on high-fat diets is what they kind of settled on. And they found several studies that presented transcriptome data about showing gene expression changes in response to high-fat diets. From there, they did some bioinformatics analysis using just some freely available websites, and they were able to do some GO-term analysis to find those that were associated with lifespan, and were able to tell something about, how diet might affect these different genes.
All right, so let's see. You answered the question about fruit flies. And
I will say something here. Sorry to interrupt. So we were talking a little bit about how to kind of make a cure your own. And what I like about just the simple techniques that I showed is that you can really cater that to do any number of different experiments. So we looked at different additives to see the effect on fertility. You can very easily switch this to a toxicology type experiment to look at the effects of different toxins in the food, and how the flies are able to survive from that. You could also see how different genotypes respond to different diets. You can look at different environmental factors by rearing the flies in different environmental chambers, and again, see the effects there. So there's a lot of things that you can do simply by changing what you put in the food or where you incubate the food, that lends itself well to doing experiments with undergrads who have very little lab experience.
Did they have any statistics before they did these experiments? Or maybe did they remember any statistics?
Did they remember? No, in both cases. So for the mini cure, most of them did not have any statistics experience. For the semester-long cure, some of them did, but most of them didn't remember much beyond a t-test. Luckily, Dr. Broach had come up with these lovely flow charts that are in the cure documents that you guys have access to, that lets them work through figuring out, based on the type of experiment that they have, what type of statistical analysis do they need to do. So that came in handy, because I probably don't know any more than the students do. I could do a t-test and that's about it. But that flow chart really helped us to narrow down what the appropriate test was.
So there's another question just about rearing Drosophila. Do you need special lighting conditions or temperature controls?
Great question. So one of the reasons I like Drosophila as a model organism is because they are easy. They don't really require too much. So for all of our experiments, we just reared them at room temperature. Optimal conditions for the flies would be 25 degrees, 12-hour light, 12-hour dark cycles. And then I think it's something like 40% humidity. You could tell, because I don't know the exact number, that that's not a huge factor for our flies, and we're out here in the desert and they do just fine out just sitting on the bench with normal lab lighting. So they're pretty forgiving for different conditions.
And I've grown them in the dark, so they will also do what you need them to do in the dark. So it's not it's not a huge deal. So back to my other question, unless other people have questions about raising Drosophila. I don't see any more questions. Your food experiment with the supplements were with first year students, and then the semester long iCure, typically students are typically juniors, sometimes they're sophomores, they're also sometimes seniors. What do you think about the question? Because we had a question that Dr. Brownell didn't get the answer. Should we be giving a cure to first year students? Should we be waiting until they're seniors for a capstone? Well, what's your thought on that?
Well, I think it's great to give the first year students exposure to research methods. In that course, I mean, we had all different majors, and there's something about, seeing a psychology major look at a fly under the microscope for the first time and be like, oh my God, that's amazing. And just see how excited they get about that. I think that's one thing that we can do to keep bio majors as well as draw other majors to the sciences, is get them excited about whatever it is that they're doing. And that hands-on experience, I think, is what's going to do it. It's not the cookie cutter labs that are going to get them really geeked out about doing science. I think it's the hands-on ownership of their project, making it personal. That's what's going to get them excited.
To me, it sounds like you think all levels are all good. I did want to bring up one thing about ASU West Campus, which is a little bit different than the Tempe campus, in that the majority of students in our upper level classes and some of our majors are transfer students. And so, what's sort of the idea then, because Sarah brought up, you have to be careful, because if most of your students are transfer students and you're giving them this amazing opportunity in their first year, then many of them are missing it. So what's the balance of that? Any thoughts on that? And it looks like Jennifer Broach might want to weigh in.
Well, I posted the demographics of our cures because I think just hearing that we're from Arizona State University, for those of you that aren't local, it seems like we're this major university with this very different background. You're like, oh, this doesn't compare to me, but if I actually posted the breakdown of how. So overall, we have 64% of our students are Pell Grant eligible, and 46% of them are first generation students. So these are students that are the ones that are working and are the ones that are needing those extra time and needing that extra that extra class. And so luckily we've been able to, with the with our partnership with the community colleges, is start cures. They've started instituting some cures there. So it's been really helpful in getting them involved early before they come over. And as well with Dr. Marshall, other grant with the STEM, where we're encouraging transfers in, and we always encourage them to do these experiences. And honestly, I mean, the population of our cures is different than the ASU population at large. So, I mean, the ASU population at large does not have that many Pell Grants. It is significantly lower in first gens. So the population that we're actually using the cure to is a less typically advantaged group.
What I think is interesting about that, based on the way I've been advertising the cure, is I have not been preferentially advertising the cure with one population or another. Indeed, I actually advertise the cure, send it off to the Barrett Honors College, I send it off to STEM, we advertise through the advisors, and they send it out to everyone. when we were in the before times, because this is unprecedented. You didn't think you'd hear that, did you? So before, we would put flyers up, we would go into classes, and we would talk about the class, and I would also advertise in STEM. STEM is a scholarship program funded by the National Science Foundation, and it is for students with need who are meritorious. And so we would also advertise with our STEM program, and we would advertise through our TRIO student support services programs. But we were advertising just everywhere because we just thought it was such a great idea and such a great experience. So we were not targeting particular populations, but yet somehow we were able to have a significantly different population than the typical ASU students. Any ideas why that is?
I do not have a good answer for that. My guess is that our population largely reflects the population that's surrounding the West Campus, more so than just the greater Metro Phoenix area. I think that plays a large role in it, but aside from that, I don't know.
So one idea that I had, Pam, is it maybe some of the biases that we've already talked about. So you have these students who are unfamiliar with the unwritten rules of how to do research, but they are go-getter type students, right? So they know they want to do research, they don't know how to get it, and then they hear about our class, boom, they're there. And so this might be one of the ways that the class really does, I think, work against the kind of biases we've been talking about all day in terms of our traditional professor-mentor model of undergraduate research.
Yeah. So I see a comment in the chat about that the timing of the research experience might have a different impact. So a freshman student versus a junior or senior, and I definitely agree with that. If you have a first year student, they're probably still trying to decide on their major, trying to figure out just where to go within their educational career. Whereas if you're looking at a junior or senior, they're thinking post-graduation. What can I do for a job? Do I want to go to medical school? Do I want to go to graduate school? And so they're more looking at that experience to drive their career, as opposed to the first years that are more exploring what they like and don't like. So yeah, I definitely think they're looking for different things. Since I had a little bit of everyone in my cures, I pretty much taught assuming they knew nothing about flies and had never done research. And so they all read the same core literature, and we walked them through what the different things meant so that they were all on the same page when they started to form their own hypotheses and look at their own literature. But I tried to play it equally for those who were from ASU and had gone through the ranks at ASU, and those transfer students that may have had a very different experience. I didn't want to assume, therefore I just assumed that they knew nothing about it, and we started at the very beginning.
So we have an interesting discussion in the chat that says, I think the timing of the research experience might have a different impact. I can imagine having a first-year student do a cure might impact self-efficacy, while using it as a capstone experience might work to verify a career choice, like you want to go to graduate school. And I think that that could be one of these backwards design questions. And then we've got some, I agree, useful to have mini cures in the first year classes and full semester classes later on. And there was one question that was in the chat with our keynote that we didn't get to that I thought we might talk about now, which is, what is more important? Is there more important the learning outcome or the career outcome? Discuss.
Well, I think it again goes back to what stage of their educational career they're on, right? I think the learning outcomes are just going to be very different depending on the population of students that you're looking at.
if I may, not only the learning outcomes different, but it depends on the strategy you want for the course. So to go back to this question of, you know, where in their education and what the purpose is, when I taught the first year biolabs, I used a very small mini cure, a 2-week mini cure. And my pedagogic goal there was actually to introduce the students how to do literature searches. But just telling students to do a literature search generally puts them to sleep. So I put it in the context of, you have to design an experiment to assess, it was bean beetle fertility. If you're familiar with bean beetles, there's actually a whole set of curricula online about using bean beetles in teaching labs. So this is another way you can employ a cure, and I think they should be at this small level employed in the very, very first classes students get into, because these are the kinds of experiences that engage and make the students interested. And if we want to talk about retention, if we want to talk about keeping people in the sciences, that's only going to happen if they are engaged with it. Nobody likes the cookbook approach.
Sorry about that. Didn't get my unmute to work. So I agree with what Ken and Jennifer have said. And the other thing I wanted to add is that whether you focus on a learning outcome or a career choice may depend on which class you are presenting the cure in. So I teach a Polynesian biology course, or cure, excuse me, but I will be utilizing that in my Gen Bio for allied health majors as well as environmental biology. So in my Gen Bio, I don't expect that to necessarily affect career choice, whereas in my environmental biology course, the outcome may be different.
Jennifer Broche?
The other thing is, it's not necessarily, I mean, I know we're talking about career goal, I mean, learning outcomes versus career outcomes, but there's a lot of those that kind of span that gap, like whether we're learning for specific course requirements or career requirements. Like there are certain outcomes, like for example, in statistics, we really emphasize, and in all of sciences, I'm not saying in just statistics, but the communication of your results. So even a mini report or a mini presentation, even if it isn't a mini cure, that communication of results goes beyond the, I mean, it includes both learning outcomes and career outcomes, because that's something that will really help you get a job, help you work as a team in the future. So there are some that kind of cross that gap, and maybe, but they're right. I mean, maybe you're not informing career choice necessarily, but like learning outcomes for helping them get a career or helping them choose a career are slightly different. But I think the communication part is one of those kind of crossover learning outcomes that are really important.
I focus on the scientific method a lot in any class that I teach, because I try to explain to my students, if you want to be a human healthcare maintenance provider, doctor, physician assistant, dentist, all of that, I hope you know the scientific method, because you should be off looking at the literature and doing data-based approaches, as opposed to just, oh, hey, I saw this dude on YouTube with this video, I thought I'd do that instead. So I try to, emphasize the scientific method. Let's see. The update of that was, I am not talking about career choice. We have tons of biology students. We struggle to place them. How important would a cure experience be for a non-science major versus a STEM allied health student? I think Jennifer Foltz, do you want to talk about that, or?
Sure. So in my opinion, a cure is equally as important for non-science majors as it is for STEM majors. So my environmental biology course is non-science majors, but a cure really gets my students interested in the course, interested in the concepts, and also it's a very good way to teach them about the scientific principles, which are a main focus in my course. So even though they're not going into a scientific field, I think that being able to ask questions about, the natural world, analyze data, things like that are useful no matter which profession they are going into.
So I want to get back to Michael's comment about not talking about career choice, but more is it about getting them a job and into professional school? How would a cure help with that? And I think a lot of the things that we tackle in the cures are things that show up really nicely on a resume. They could talk about how they designed a specific experiment to test, X, Y, and Z. They'll have a poster that they can actually show off, like, look, this is what I did. At the end of the course, they should be able to say, they should be able to talk about their project. Most likely they have presented their project to their peers as well as faculty members, so they know how to talk about their project and answer questions about it. So they really, again, they own their project, and that is a great selling point when you're talking about any profession is, look, I did this project, I developed it from start to finish, I worked with people outside of my field, we were able to generate a successful collaboration, and look, this was the end product that was successfully presented to all of these different groups. So I think there's a lot of things that can cross over, not just to science majors, but to all other majors as well.
Great. There was one technical question in the chat a little while ago about the core literature. Approximately how many papers are they reading for the core literature, and how much time do they have to read it? And I believe in our documents there are 2 examples of some core literature lists in there. So there are a couple of examples, but how many papers did you assign, and how long did they have to read them? I think for me, it was when I taught it in the semester, it was 3 or 4 papers, and they had a week. But I specifically I was very deliberate in the papers that I chose, and we talked about how to read a paper and what I was looking for them to read the paper before we gave them the core literature. I didn't just say, hey, read this, it's like, here's Kafka, read it, right? It's forget it.
Yeah, same. So I was very deliberate in the papers that I chose as well. They had 3 articles for the full semester cure. They had, since we met twice a week, they had 1 paper for each day. So 1 Tuesday, 1 Thursday, and 1 the following Tuesday to go over. And I kind of arranged them in increasing order of complexity. And then we would talk about them. So that everybody was sure that they understood the main points of it. And I tried to emphasize the main points of the article so that they wouldn't get bogged down by all the technical jargon that was often there, because I think sometimes that just freaks students out. But I think starting with the simpler, just kind of introductory paper was a good way to just get them started with literature, and talking about it in class further just helped to solidify some of the concepts.
So in my example, so the first full semester cure I did was actually on capsaicin content in hot chili peppers. And I gave them about 6 papers. And I also used it, amazingly enough, to what I hope to help build students' own confidence. So I started it out with the 1912 paper from Scoville himself, describing Scoville heat units, so that I could also show the students where science has come from, right? That's a radically different paper than what we see. Now, of course, before that core literature, I like to share with the students a paper or 2 on how to read scientific papers, so that we start that way. But with the core literature, so what I did is I set the stage, right? You have Scoville. This is how we measure how hot peppers are. We skip then to 1977 when a scientist named Todd decides to stop using the human tongue and use gas chromatography. So now we get the modern way we measure this. Then I have a collection of papers that talk about different attempts at manipulation of capsaicin levels or other parts of agriculture with peppers, but basically examples that I think have experiments in them that are really good examples for what the students could accomplish in a semester. So that was my strategy with the peppers. And yeah, there was a lot of subterfuge there, if you will. You know, it is that chance to introduce kind of the history of science and development of science and put a more storybook phase to it, so it's not all technical. But that's how I constructed my core literature.
So I was looking to really show the versatility of Drosophila as a research organism when I selected my my 3 articles. So the first was more of a general overview about, you know, this is why flies are so awesome to use. And then I selected another paper that looked at different techniques to study fly fertility and some things that they found when they manipulated diet. And these were all, you know, laboratory experiments. And then the last paper was more of a bioinformatics paper to show how you could study the same type of thing using fly information and do this as a totally in silico project as opposed to doing wet work at the bench. So we kind of had all of the different aspects of flies covered.
Yeah, I think for each project, you really, at least for me, you know, I wanted to shower them with 20 papers, and this is the most interesting paper, and this is the most highfalutin technology ever. And then you have to balance that with what are they capable of reading and what are we capable of doing in a semester, you know, with a smaller budget perhaps. And you know, we have an XRF. A lot of places don't have an XRF, but you know, there are other things that we don't have that maybe either a larger institution has, or I mean, we have core facilities, but you know, I couldn't pay for, you know, 20 hours on a ICP-MS or something like that. So I think there's a real balance there. Well, you know what? I can't believe it. This day is almost complete. And hopefully everybody has gotten some ideas and some information and some takeaways. And we put into the chat the link for the documents. I am going to go ahead and put Dr. Brownell's slides in there so that you can find those slides. We did send out an email earlier today with a Zoom link for those of you who registered. And so don't forget to come to the Zoom link. And don't forget that we are going to be sending you surveys. We'll be sending you a survey this evening and at 3 p.m. Our time, 3 p.m. our time, not in the past for your time. And we hope to see you in the Zoom, and I will send another email tomorrow morning with a new link for a live YouTube then. And so it's going to be a different link than this link was today, although I think you could just go to the YouTube channel and find us as well. All right, so we will hopefully see you in about 15 minutes in the Zoom. We're going to take about 15 minutes to decompress, and we'll see you. And any last words, anyone else? All right, I guess may the cure be with you. I think I'm funny. Okay, see you.